Thanks very much for having me. Today I'm going to tell you about the use of CRISPR technology as a tool for understanding and treating FSHD. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is a bit of a mouthful. But essentially this refers to a primitive immune system which was developed by bacteria that allows the recognition and destruction of foreign DNA. So basically a form of antiviral defense for bacteria. And in 2012, it was recognized and developed as a tool for editing the genome of virtually any species. It's important to point out that CRISPR is not the first gene editing technology, but it is by far the most versatile and easy to use, which has led to the explosion of interest in recent years. And the practical applications of this technology are numerous. It's already been widely used as a tool for basic research. But what really excites people are the many uses of CRISPR for biotechnology, and what most of us are especially interested in, the potential to cure or treat diseases. So what exactly is CRISPR and how does it work? The system has two components. There's a protein called Cas9 and a strand of RNA, which is shown here in green, that binds to Cas9 and guides it to any matching sequence in the genome. Cas9 is a protein that cuts DNA like a pair of scissors, and it will cut the genome at any place targeted by the guide RNA. And in some cases, you can just cut out the original DNA and allow the cell's own repair pathway to fuse the broken ends. But you can also provide a donor DNA template, which can be any desired sequence, and another repair mechanism in the cell will insert the new DNA in place of the old. And having used the scissors comparison, I'd like to call your attention to this article called "Why the Molecular Scissors Metaphor for CRISPR is Misleading," which was published on The Conversation. It's written by a scientist named Eleanor Hordle, who does a fantastic job of really putting the CRISPR tool in the context of a living cell, within an organ, within the entire body. It's just a short article, but it really gives a great sense of the complexities involved in using CRISPR as a genome editor, and also the complexities of any medical treatment that perturbs some target within living cells. It's very, very much worth reading. Oops. So what makes CRISPR such a powerful tool for therapy is that it really gives us the potential to permanently correct the underlying cause of a disease instead of just treating symptoms. And because of this, many CRISPR startup companies have been launched in recent years. These are just some of the more prominent. And of course, there's a huge projected market for this. So people are taking this technology very seriously. So what's been done in terms of using CRISPR for muscle disease? A number of groups have been working on using CRISPR for treatment of Duchenne muscular dystrophy, which is caused by mutations in the dystrophin gene. And a couple of years ago, 4 independent groups reported Cas9-mediated gene editing in a mouse model of this disease. They showed that they could restore dystrophin expression to therapeutic levels in the affected muscles. And a couple of years, or actually more recently, this year, 2 groups showed lifelong correction in the mouse model with no major adverse effects. And there's even been a proof of principle study in a dog model of the disease, showing that dystrophin expression could be restored in the affected muscles. So this is very promising, but the question for us is, can we use CRISPR technology as a therapeutic approach for FSHD? So just a brief overview of what causes this disease. It's well established in the field now that FSHD is caused by misexpression of the DUX4 gene in skeletal muscle. So the disease locus is this large array of repetitive DNA on chromosome 4. About half of our genomes are actually composed of repetitive DNA. And some of it is functional, but a lot of it is dangerous and needs to be silenced. And in the healthy situation, there are silencing proteins that bind and modify this region to keep it silent so that no genes can be expressed. In FSHD1, there's a reduction of the array. So instead of having hundreds of these repeat units, there are only a few. And this causes the DNA to lose its repression and become active for gene expression. And now the DUX4 gene is expressed from the very last unit of the array. FSHD2 patients have normal-sized arrays, but there are mutations in these silencing proteins, and the end result is the same. The DNA loses its repression, becomes active, and DUX4 is expressed. So why is DUX4 so bad? It turns out that the DUX4 protein actually has an important role early in development. When a fertilized egg reaches the cleavage stage, DUX4 turns on a program of gene expression in these early embryos, which is important for getting them to the next stage. But after this point, it has to be shut off. And when DUX4 is misexpressed in adult skeletal muscle, it activates an aberrant program of embryonic gene expression, which causes muscle pathology and cell death, and eventually results in the disease. So what are the potential uses of CRISPR technology for the treatment of FSHD? Well, everything I've talked about so far is standard CRISPR editing, where a guide RNA recruits the Cas9 protein to a specific location in the genome, and Cas9 makes a permanent, irreversible cut. This cutting system is what the vast majority of researchers are using to correct diseases. But it's extremely inefficient in most cases, which is one problem. And it's also not an ideal system for FSHD because the disease locus is just one repeat with hundreds of nearly identical repeats in the genome. And cutting your genome in hundreds of places is really not a good idea. But an alternative system is CRISPR inhibition. And in this situation, Cas9 has been mutated, so it's no longer capable of cutting, and it's fused to a repressor protein. So now the guide RNA recruits a system which represses DNA instead of cutting it. And this turns out to be really ideal for FSHD because we want to turn off or dial down DUX4 expression. And importantly, other similar repeats in the genome are normally repressed. So recruiting a repressor to those locations shouldn't cause any problems. If the repression is stable enough, it can be inherited by subsequent generations of cells, but importantly, without creating any permanent changes in the genome. So in 2016, we published a proof of principle study showing that CRISPR inhibition can in fact reduce the pathogenic gene expression in FSHD. So using cells from an FSHD patient, muscle cells, we showed that the system returns the FSHD locus to a more normal state of repression and significantly reduces the expression of DUX4 in its target genes. But of course, correcting cells in a dish is one thing, but actually therapeutically delivering these components to a patient's skeletal muscle is actually quite challenging. For some diseases, blood disorders, cancer immunotherapy, you can actually take cells out of a patient's body, you can modify them and expand them in culture, and then you can introduce the corrected cells back into the patient. But muscular dystrophies require targeting of essentially all the skeletal muscles in the body. And one way to accomplish this is using AAV, which stands for adeno-associated virus. This is a virus that's been heavily modified so that it's safe, and it's essentially used as a vehicle for delivering therapeutic genes. But one problem with this vector is its small packaging capacity, which means that very large genes won't fit into it. But this is actually a great example of how muscle disorders all share common challenges. So progress in any of these diseases really benefits all of us. So it's true that large genes can't be delivered using current AAV vectors, but there are ways around this challenge. And what you have to play with is this therapeutic cassette, which consists of the actual components you want to deliver and regulatory elements which drive strong muscle-specific expression of these components. And one thing you can do is to reduce the size of this regulatory DNA, as Steve Hauschka and his lab have done very successfully over the years. You can also reduce the size of the therapeutic gene, as Jeff Chamberlain has done by creating smaller versions of dystrophin for Duchenne muscular dystrophy treatment. And for CRISPR platforms, you can use DNA encoding smaller versions of Cas9 from different species of bacteria. So there are ways around this delivery challenge. So what are some other challenges for CRISPR technology as a therapeutic avenue? There are some general challenges which apply to all diseases. So we talked a little bit about delivery. Even if the therapy is delivered to all the target cells, would all of them be modified? What happens if the CRISPR system is recruited to other places in the genome? This gets at the idea of off-target effects, which are a serious concern. Can CRISPR components stimulate an immune response? Remember, Cas9 is a bacterial protein that the human body is not used to seeing. And of course, will the CRISPR system actually provide a long-term cure? And we believe that it will, but we won't know for sure until we try. There's one very serious concern related to using CRISPR and its cutting capacity, and that's that in certain situations, cutting by CRISPR can result in edited cells that are prone to become cancerous. But importantly, this would not be an issue with CRISPR inhibition. And then there are some FSHD specific issues, which are things we still need to learn about the disease, like how many muscle fibers actually need to be corrected. Do muscle stem cells need to be corrected? And exactly how much of a reduction in DUX4 expression will really provide a functional benefit. And regarding that last question, I think it's worth emphasizing that in the healthy situation, the disease locus is heavily repressed by multiple mechanisms. This is a region that really wants to be off in most adult tissues. And we know that some asymptomatic individuals, so these are people who are genetically FSHD but show no signs of the disease. And some of these people express low levels of DUX4, which suggests that we don't need to turn DUX4 off completely. We just need to reduce it to asymptomatic levels. So even small decreases in DUX4 expression should have some therapeutic benefit. And just as a side note, I want to mention that CRISPR technology is also useful as a tool for FSHD research. So far it's been used to identify regulatory factors binding the disease locus by Steve Tapscott's group. They used dead Cas9 fused to a tag, which allowed them to isolate the DUX4 gene with all of its associated proteins and then identify these proteins. We've also used CRISPR inhibition to validate knockdown of candidate DUX4 regulators. So in this case, we were not targeting DUX4, but rather genes encoding activators of DUX4 expression. And it's also been used to correct the FSHD2 mutation in muscle cells in culture for testing the rescue of disease phenotypes. So in addition to its therapeutic potential, CRISPR is also helping us to better understand the disease that we study. And I just want to end with a word about the state of CRISPR in clinical trials, which have moved slowly in the US and in Europe due to very legitimate concerns about safety. But there's now a trial for a CRISPR-based therapy to treat a form of inherited blindness. This would correct the mutation in a photoreceptor protein, and it would be a local treatment, so patients will be injected under the retinas. And importantly, with this treatment and all the others, these are treatments of somatic cells, which means we are not altering sperm or eggs in any way. No alterations of germ cells. These are changes that will not be passed on to offspring. And this treatment was developed by Editas Medicine in Cambridge in partnership with a pharma company in Ireland. And CRISPR trials for cancer immunotherapy and beta thalassemia are already in the works. Trials for sickle cell disease and others are coming. So as a therapeutic approach, CRISPR is still in the early stages, but it's potentially very powerful and very exciting. And it's important to remember that the platform is constantly evolving and being improved on because there are a lot of people working very hard on it. So I just want to thank our lab's funding sources, especially FSHD Canada, Chip and Shannon Wilson, FSHD Global, Chris Carino, and the FSHD Society for supporting our CRISPR work. So thanks very much. We have plenty of time for questions.
Thank you so much. That was a great presentation and a great update on previous webinars that you've given us. I invite the audience to start posting questions in the Q&A box. Just click on that Q&A link underneath and a box will pop up and you can just start typing in questions. So first question, I guess you covered this somewhat, but they were interested in understanding the delivery mechanism of the potential therapy. So you had mentioned AAV, there might be others, but would you like to elaborate on maybe some of the, why AAV and what some of the concerns and challenges are?
Sure, so let's go back. Yeah, so you see another delivery mechanism here is lipid nanoparticles. Those are also being developed as sort of a non-viral mechanism. People think in many ways this might be safer, even though AAV has been shown to be relatively quite safe, and is being used for gene therapy of liver diseases. These are still in the early stages of development. I think right now people are using them to target hepatocytes in the liver and maybe the kidney. Yeah, the problem with these is they just don't go efficiently to the organs in which you want them to go to. AAV, on the other hand, is great because there are different serotypes of the virus that have tropism for different organs. And so, for example, AAV6 and AAV9 are highly tropic for skeletal muscle, which is perfect for FSHD. So really the big challenge with AAV, of course, it would involve an injection. But the big challenge with AAV is the packaging capacity. And as I mentioned, there are ways around this problem, which I'm working on, and certainly other people in muscle disease fields are working on.
Right. Are there also concerns about AAV eliciting an immune response in the part of the patient so that they might not be able to have multiple doses? Is it a one-time thing where once you've taken this therapy, you have an immune response to it?
Yeah, I think that's the current state. I'm not not as up on gene therapy as I should be, but yeah, that's also a concern. But which is why things with CRISPR inhibition, if there are ways to stably turn off DUX4 that can be inherited by subsequent generations of cells, that would be ideal, because then a single delivery could in theory result in lifetime repression. We would hope. Yeah, but of course, the gold standard for delivery is not things, the way CRISPR can be delivered. What you really want to take is a drug. And, those will be the first clinical trials for FSHD will be, orally available, compounds. And I'm sure, we'd all rather swallow a pill than, than anything else.
Right. So the question is, this sounds very promising in animal models. How long before this is available in humans? So you mentioned it's already in clinical trials for very specific indications. I guess let's make it more specific for muscle disease. So what's the development path look like?
Right. And part of the complications are shown here. These challenges are things that are particularly challenging for a muscle disease and less challenging for a blood disorder or a cancer immunotherapy, where you can take the cells out, you can modify them, put them back in the body. So if we look at this CRISPR-based therapy, which is, they're now recruiting patients to treat this Leber congenital amaurosis, this form of inherited blindness, it's really nice because the eye has been sort of traditionally considered to be an immune privileged organ. Certainly should be less of an immune response by injecting the eye as opposed to systemic injection. And also, it really is a local treatment, just an injection under the retina. So they certainly don't have to target, multiple muscles, across the body. So yeah, it will be for these easier diseases that are easier to treat, easier to target. And these will be the first thing to get to clinic for CRISPR therapies, definitely. So yeah, I can't say exactly how many years, whether CRISPR will be in the clinic for FSHD. I hope so. And we're certainly going to, our feeling is always that the first things to make it to the clinic are not always the best treatments. And it's never good to abandon all lines of, all avenues of therapy because you just never know. Look at myotonic, they cured the mouse and the clinical trial failed. And you just never know. Mice are not people, and the only way to really know it is to take that leap of faith. But you never want to take that leap of faith until all of the proper, experiments have been done. The data is believable, it's consistent. All the safety studies have been done. So once you've done the due diligence, then it's time. But so we're going to keep working on our CRISPR approach as well as other approaches. But the great thing about the last 10 years is that we have learned so much about FSHD in the last 10 years that, now we really do have a disease candidate. We have fantastic therapeutic targets, and everyone in the field is focused on something different. Everyone is thinking about therapy in a different way. And I think having those multiple points of attack can only be a good thing.
Great. So maybe going a little more in depth into some of the animal models that have been studied. I guess it wouldn't be for FSHD yet, but there's been a lot of work in using CRISPR to try to treat Duchenne in animal models. Can you comment on what those models have actually shown so far in these, for Duchenne? Is it, have they recovered function, or is it just showing that CRISPR can actually repair the dystrophin protein, or
yeah, so
what is it actually showing?
Right. There are quite a few papers. These are the more prominent ones.
Right.
They really do show that. So the nice thing about Duchenne, is that you don't have to recover full normal levels of dystrophin to get therapeutic benefit. So yeah, some of these studies have shown functional benefit in terms, various strength tests. But the levels of dystrophin that are actually recovered are highly variable, the small percentages in many cases. The other thing about Duchenne is that they have to definitely have to get dystrophin expression back in the heart, which these studies have shown. FSHD, that's not a challenge. Luckily, we only have to target the skeletal muscle. The important thing is really this lifelong correction, showing that long term with no major adverse effects. Again, the potential for edited cells to become oncogenic is something that has come out recently, and people are saying, well, haven't they seen this in mouse models? And the question is, have they seen it and not reported it, or have they not bothered to look? And that's really not for Duchenne, but for other uses of CRISPR. And luckily for Duchenne, the repair pathway in the cell that can be used for CRISPR treatment of Duchenne is not susceptible to that. So that would be, let's see if I have it here. Yeah, so this NHEJ pathway, which creates small insertions and deletions at the point of the double-stranded break. This pathway operates just fine, and there should be no problem with cells that are edited by this approach being oncogenic. This is the pathway that people are concerned about. So luckily that's not an issue for Duchenne, but yeah, again, I always bring up the example of myotonic dystrophy. The cells really were cured. It was a wonderful model of, but how good can a model really be if it fails in patients and, mice are completely cured. But we have to do the due diligence. We have to use the best models we can to answer the appropriate questions and before starting clinical trials, especially for a novel technology like CRISPR.
Right. And it goes back to your point. It's important for people to take many approaches and to do that work really well.
Yeah.
They have clean data, and you can say this idea is
right.
Promising. This idea just flamed out, but we know why.
Right. Yeah, exactly.
So, yeah. So there's a question here. Let's suppose that CRISPR treatment is effective. We're able to shut down DUX4 in skeletal muscle in patients. If that were to be accomplished, would regeneration be possible in those muscles that have been affected?
Yeah, I would actually think so. I mean, this is just me speculating, but I would think that especially in the early stages of the disease, you should be able to recover a lot of function. So it wouldn't just hopefully stop the progression, but actually you should be able to restore, healthy muscle. Of course, in the late stages of any dystrophy, I think that becomes much more difficult. But yeah, I think there's absolutely reason to think that would be the case.
Then there's several questions having to do with you mentioning that you don't necessarily have to shut DUX4 expression off completely. How do you know what amount is enough to achieve a therapeutic?
Yeah, we really don't. The best I can say is that the fact that these asymptomatic individuals do have detectable DUX4 expression tells us that, yeah, a little bit is okay. It's not going to be a problem. But of course they have less than their manifesting relatives. So yeah, I mean, that really gives us hope that, we don't have to completely knock down, just as Duchenne patients don't have to recover full dystrophin expression, hopefully FSHD patients won't have to completely obliterate DUX4 expression. And yeah, there are so many repressive mechanisms and some activating mechanisms, which these active mechanisms of aberrant DUX4 activation and FSHD are great drug targets. So these are great targets for small molecule inhibition, which we and other labs are also working on. So yeah, multiple approaches and reason to believe that we can knock down DUX4 and actually have a benefit.
So here's an interesting question. Are there approaches that involve not eliminating DUX4, but perhaps changing it in some way so that it's no longer able to do damage?
Huh, that's interesting. I don't know if anyone is working on changing DUX4. It becomes one of those things where, sometimes I think to myself, especially after reading that article I mentioned by Eleanor Hortel, that it's really incredible. Living systems are so complicated, human beings especially, and it's just incredible to me that we can ever mess with a living system and come out ahead, because things are so complicated. And, it's just so much easier to screw something up than it is to fix it. DUX4 is one of those things where the function, the primary defect occurs upstream of DUX4. It's really at the locus, at the disease locus, where the repression is lost. And, because of that, DUX4 is expressed, and then DUX4 has all of these downstream effects. It turns on all of these genes, which are only supposed to be on in germ cells or the embryo. And those have all these pathological effects. So, some people are focused on trying to cure the downstream pathology. And the problem with that is we're not really sure which of those pathways, is actually relevant in terms of the pathology. Are all downstream effects of DUX4 pathological? We don't know. Some of them probably are, but which ones are you going to attack? It seems better to me to look more upstream and try to cure the problem at its source, which is what we've tried to focus on. So messing with DUX4 in some way, I'm sure there are ways to do it, but it's like anyone who takes any kind of medication knows there are always side effects, and there are side effects because drugs are not specific. Drugs in general bind to some protein groove which looks like other protein grooves, and you have these off-target effects. Also, proteins don't do just one thing. Every protein in general, DUX4 may be an exception, but most proteins have multiple functions in the body, and it's very hard to mess with just one of those functions without perturbing all of the rest. Luckily, DUX4 is something that really does seem to be only important at this very early stage of embryogenesis, and also in the testes. It seems it's highly expressed and probably performs some important role in the adult testes. So but it's a good question. I'm not sure anyone is doing that right now.
So I have many questions asking how soon before this can be, I guess, in clinical trial, let's say. And so if you were to walk back and say, well, where you are with your research now, what needs to happen before it can be, before you have the data you need to be able to,
Yeah.
say, we're ready to go into the clinic. What needs to happen?
Right now I'm working on basically this, using a smaller version of DCAS9 fused to larger repressors. And hopefully the repressors we're using will provide more stable repression. We're going to test them in our animal models of the disease. We have a mouse xenograft model with Bob Block that he developed, and also an FSHD model of the disease in the lab. And we will test our best CRISPR system in these mouse models. And after that, I don't know really how much more we can do if that data is promising. So yeah, hopefully within a few years, CRISPR for FSHD will be in the clinic, in a clinical trial. But we'll see. In the meantime, small molecules are coming rapidly. So we're not rooting for one or anything that works well and quickly would be fantastic. So.
Okay.
Yeah.
So great. Yeah. So I would say say things go fantastic, you've got your kind of modified CRISPR construct that can be packaged into AAV. And in the meantime, there's also a lot of work going on to show how safely AAV can be used, perhaps in a Duchenne situation or one of these others.
First, yeah.
It would probably have to be in a muscle disease because we're, you know, you're looking at huge level of exposure of the body to the AAV gene, not just the eye, not just the liver. And say that goes well, then when you begin the clinical trial process, there's going to, I assume, have to be quite extensive safety testing still, because this is a new, you know, completely new approach, and that might take 1 to 2 years just to make sure it's safe, and then another 2 years to do a phase 2 trial, and then we'd want to know, you know, and in the case of FSHD, it might qualify for what's called an accelerated approval, where because the disease progresses in a slow and somewhat unpredictable way, it's very hard to prove in a, say, a 3 to 6 month time frame of a clinical trial that it has actually, the treatment has actually stopped the progression of the disease. So the FDA in those sorts of circumstances might approve a drug, assuming they feel it's safe enough. They might approve it on the condition that you continue to monitor every patient who takes it beyond that point. And within a time frame of something like 5 years, you still need to collect data to show that it's effective. So that's the kind of time frame, I think, you know, generically speaking, that we're looking at for clinical development.
Yeah, thanks, June.
Okay.
More about the development process, but just in terms of the basic science that precedes that, I would say a few years for sure.
Right. So I'm curious, have you been approached by biotechs that are interested in this application of CRISPR for therapy development? Are you seeing interest in?
Yeah, we are definitely. Yep, and we've spoken to several companies who are developing their own CRISPR platforms, and you know, a lot of people are developing, especially companies. You know, they got the technology, they need a disease to go with it, and FSHD is a really perfect disease in many ways for certain CRISPR approaches. So no, it's the interest has been very very encouraging. It's really nice because this is something we can't do by ourselves. You know, this really takes a huge partnership between academic scientists, industry, and pharma, and patients and their families. It really takes a lot of people to make it work, to actually get a therapy into the clinic.
Right. So related to clinical development, there's a question about whether this kind of treatment would be available to children with infantile onset FSHD.
Well, I would assume that if CRISPR makes it to the clinic for FSHD, in adults, that I don't know regulatory differences between, whether there are certain other safety things that have to be tested or implemented. But just from a basic science standpoint, thinking about what we're doing to the locus, I would say, there's absolutely, it should work for the infantile form as well as the adult form.
Right. And, with a drug, like a medication, often children metabolize drugs differently than adults. So there's another layer of safety testing and kind of a higher bar for doing clinical trials in children. But it's going to be very likely that Duchenne, which is a pediatric disease, and that there may be data coming from Duchenne field just on safety of like these AAV vectors in those children. So maybe that's something that will be helpful for advancing it for infantile FSHD.
Yeah, that's great.
Yeah. So, okay. There's so many questions. Oh, okay. Here's a question that probably someone picked up on your comment about CRISPR being something that can be passed down from one generation to the next. And in this situation with FSHD, you were saying you're using an approach where that won't happen, because one of the big worries is that you introduce a change in the genome and then it is perpetuated, it stays in the species forever. But I suppose from the perspective for a family that has FSHD, that would be a desirable outcome, that you don't have to ever worry that your children will have FSHD. But can you explain a little more about that, generational?
Sure, and maybe I hope this wasn't confusing. Yeah, so when I mentioned that the repression, using CRISPR to inhibit the locus, if this repression is stable enough, it can be inherited by subsequent generation of cells. So I meant, muscle cells, as they exist in the body are post-mitotic, so they don't divide. But when you injure your muscle generally through exercise or any other kind of muscle injury, then the muscle stem cells actually are activated, and those are what give rise to new muscle. So in cases like that, if you were to, basically repress the DUX4 locus in satellite cells, they should be able to divide to give cells that also have the repression. But yeah, in terms of being inherited by people, yeah, absolutely. We are only making changes in somatic cells. Muscle is a somatic cell, it's not a germ cell. So in order for any CRISPR change or anything to be inherited genetically, it would have to be, the alteration would have to take place in the germ cells. And yeah, there are just too many concerns about that. So I don't think anyone is considering that approach, at least in the US or in Europe.
Okay. So here's a question. Your theory seems to make an assumption that further repression of already repressed DUX4 won't cause harm. Is there a version being developed that only represses the one on, I guess, the 4QA haplotype, so and leaves others alone? So
DUX4 really is not expressed after, where is it? Yeah, after this cleavage stage. So, for treating adults, this really isn't an issue because DUX4, as long as we don't target the germ cell, the testes, where DUX4 still seems to be expressed and probably does have a role in the adult, although that hasn't yet been studied. If we use, for example, an AAV that really targets skeletal muscles, it really should not go to the testes and cause any problems. It's a good question, yeah, luckily this is a protein that really shouldn't be expressed at all in adult tissues. So turning it off really should not cause any problems.
Mm-hmm. So here's combining a couple questions. Someone was asking about any known major side effects of using CRISPR. I guess it probably has more to do with the AAV, the virus that's used to deliver CRISPR. There's some concerns, I suppose, with side effects there. And then kind of related to that is, once that's introduced into the body, it's really difficult you can't reverse it. You can't take away, you've put 1 trillion AAV cells in the body, you can't suck them all back out and take it back, right? So what are the concerns around potential side effects and irreversible?
Yeah, so these still need to be studied, really, particularly with CRISPR, so the questions of the off-target effects, get at side effects, immune response, I mean, that's another side effect that would be really unfortunate. So one thing people are doing, I don't know what the current state, but relatively recently, scientists have been looking at introducing CRISPR Cas9 inhibitors. So these would be something where you would basically apply the CRISPR treatment, and then afterwards you would basically inhibit CRISPR to knock it out and prevent, hopefully, the off-target effects which might occur later. So there people are working on ways to make Cas9 less immunogenic by altering the protein structure. And there are also other CRISPR-like systems, you know, that are similar to Cas9, do the same type of thing, which are less immunogenic. So, you know, these are all things that people everybody's worried about. Everybody is working on ways to solve. But yeah, it's a good question. Side effects, always side effects.
Okay. Here's a question. Did I correctly understand you to say that the problems of modified cells becoming cancerous was not as great in FSHD?
Oh, no, sorry, I must have misspoke. In some ways, yeah. So the system that we're using, the Crispr inhibition, will not be, this will not be an issue. This is a problem with using CRISPR as a cutter, using it as the scissors to basically cut DNA. And that's because CRISPR a couple of studies found earlier this year that CRISPR basically stimulates p53, which is something that either repairs DNA damage or causes the cells to die. And so cells that survive the editing process tend to have p53 mutations. And since p53 is a tumor suppressor, the fear is that these cells will become cancerous. So that is the fear, but it's only with one of these editing pathways. Yeah, so you see there, after the double-stranded break induced by the CRISPR cutting, there's this NHEJ pathway, which occurs in most cell types. And then there's this HDR. So this is the pathway where if you have a template that you want to, basically new DNA you want to add in place of the old, this is the pathway that you're relying on. And this is the one that where that is a concern, where edited cells may be oncogenic. This pathway, that is not a concern. And luckily for Duchenne, a lot of, the dystrophin mutations can be fixed using this pathway. So it should not be a concern for Duchenne, and it won't be a concern for us using, because we're not using the CRISPR tool to cut at all. We are using it to repress. So in many ways, this is really a much safer system.
Great. There's a question here. How different is this approach from the stem cell technology that you may be aware it's being offered in other countries? Like there's a stem cell therapy being offered in Iran, in Mexico, in China, and so forth. That's
well, it's, you know, completely different approach. I mean, that I'm not really sure how or if that works, or for how long. But this, I mean, we understand the biology. We really understand what's going on in terms of trying to treat FSHD at the root cause of the disease, target the locus specifically, and really fix the dysregulation at the locus. Yeah, but I can't really comment too much on
okay. All right. So a number of questions relate to the CRISPR field itself, like how many people are working in this field and how quickly is the technology itself developing?
Yeah, no, it's a good question. So there are a lot of people who work peripherally in the CRISPR field, like me. You know, I'm not a CRISPR pioneer. This is not a technology I developed. You know, I'm basically using it for FSHD as one of our approaches to cure the disease. But there are people who's, you know, they really are CRISPR pioneers. I mean, they're absolutely brilliant scientists who, you know, said, hey, this is a fantastic system. And it was already developed by bacteria. They did all the hard work. You know, they invented this amazing primitive immune system, and we can use it as a tool for biotech and for diseases and all these things. So there are people, and all the biotech companies, a lot of them, you know, are heavily focused on the technology, improving the technology, finding ways to make it more specific and more effective, more efficient, you know, and like I said, a lot of them are looking for diseases to give them some kind of, you know, reason to improve the technology. So this is where the partnership is really important. But yeah, there are a lot of people. I mean, I don't know exactly how many, but, you know, you can get a sense just from, you know, these are just a few of the startup companies that are based on CRISPR, as that's what they do, you know, and these are just a few, and they're staffed by people who really have a strong interest in getting CRISPR to a clinical setting. So yeah, but then there are a lot of researchers, you know, like me. I mean, I don't consider myself a CRISPR researcher. I consider myself a, you know, an FSHD researcher. and a muscle biologist. And you know, CRISPR is just a tool that I use that I think has a lot of promise for treating the disease. So yeah, but it's great because, you know, the people working on CRISPR are working on it across all fields. And, you know, like I said, a lot of these fields have multiple problems in common, and having other people solve, you know, some of your problems for you is a great thing. So you don't have to do everything.
Right. All right. Here's another question. It has to do with muscle stem cells. So the idea is, can you imagine harvesting muscle stem cells from a patient, correcting them in the test tube, basically using something like this CRISPR approach, and then reintroducing them into the individual?
Yeah, that's a great question. One of the problems, though, with muscle stem cells is that they are very, very low abundance. They're really, really, there's just a few of them, and they hang out kind of in the niche, you know, outside of muscle fibers. And another problem with correcting stem cells and reintroducing them is stem cells are really, muscle stem cells are quiescent. You know, they don't divide unless they're given the appropriate growth signals. And you know, there are people who believe, you know, with good reason, that just putting them in a dish is activating them. You know, you're already changing them. And so it would be complicated and difficult. Yeah, I'm not sure. Yeah, how practical it would be, but it's a great question.
Mm-hmm. Somewhat related, I guess, is whether or not you use these engineered stem cells, or whether you introduce the therapy directly into patients and transfect the muscle cells. And presumably you have to also reach the muscle satellite cells, right? These are muscle stem cells.
Yeah, that's one of the questions. We don't really know, but yeah, do muscle stem cells, you know, need to be corrected? Because, you know, you can envision correcting, you know, the actual differentiated muscle cells that make up your fibers, you know, like in your biceps. And, you know, that might be good enough because the cells don't turn over very often. You know, really, the only time you damage muscle is in response to exercise or disease. And so, you know, they're only, it might be enough. We might not have to target muscle satellite cells, but we don't know that.
Okay.
You know, and there's no way really to know until we try.
Right. There's a question here relating to the role of DUX4 in embryogenesis. So that's where you mentioned before that I think it's on the second day, I guess, of embryogenesis, or, you know, it's one of the earliest events that happens in a fertilized egg. Does this research inspire alternative treatment avenues for FSHD?
No, because I think most of us are focused on treating, you know, children, adults, not certainly embryos, and so I don't think it would be an issue. But yeah, DUX4 clearly, you know, is extremely important for getting, and this is work from Steve Tapscott and others, getting these early embryos to the next stage. It's really important, and the program that it turns on is interesting because, you know, it turns on a whole bunch of genes which are, in many cases, they're immune mediators. And so, you know, the researchers that did this work speculate that it may be important for protecting the embryo from the maternal immune system, so that the maternal immune system doesn't attack it. So yeah, it's actually what DUX4 does during development is probably fascinating, and there are a lot of people working on this to better understand it. But yeah, for therapy, it should not be a problem.
Mm-hmm. You mentioned earlier, another question, that the AAV vector was heavily modified. Would it be impossible to modify that AAV? So I think she's alluding to the fact that you said that it can only carry so much cargo.
Right.
Are people looking at ways to modify the AAV itself so that it could carry a larger cargo?
Yeah, AAV researchers in the gene therapy field have been working on this for many years. And really, I think they've got it pared down as much. It's been, this is a lot of work as we've gone into this. So these viral sequences, I think, are absolutely essential. I don't think you can pare them down anymore. But the things that they've gotten rid of are things that features of the virus that would allow it to propagate in, you know, basically propagate in a person, which would not be good. Certainly don't want viral infection. Yeah, so this part, this therapeutic cassette, is really, you know, what we can trim down a little bit. So I mentioned, you know, Steve Hauschka reducing the regulatory DNA. I did a lot of that work when I was in his lab, so still kind of playing with it, you know, and yeah, I mean, the more space you can allow yourself, you can basically give yourself the opportunity to put larger proteins, genes for larger proteins in here. And so smaller DCAS9, smaller regulation, bigger things gives you more flexibility and the potential to express more powerful repressors. So yeah, we are definitely working on that.
Okay. So I have a couple questions here asking for comment about PAX7. So our audience follows the research closely, and they've heard about PAX7 as maybe kind of a competitor, DUX4, in that kind of role. Can you comment on how that fits into all of this?
I don't think it's really, well, that research is a bit controversial, and I'm not sure that it has much relevance regarding CRISPR therapy. We would definitely be affecting DUX4, not PAX7 expression. So yeah, it's not something we're concerned about.
All right. Do you anticipate that a CRISPR-based therapy will benefit people, seniors who've had the disease for a long time?
Yeah, I think, you know, like with most things, it's going to depend on how far advanced the disease has gone. You know, in the late stages of disease, there are some, you know, people in their 60s and 70s who still have very mild form of the disease. And in those cases, you would hope that, yeah, that they are amenable to treatment. You know, and of course, if the dystrophy's progressed, you know, very far, even in a younger person, you know, it may not be as effective, the treatment. So yeah, I would think that, you know, even, yeah, older patients, if they don't have a very rapidly progressing form of FSHD, the CRISPR treatment should, you know, should certainly help them too.
Okay. So theoretically, if we can use CRISPR to suppress DUX4, I guess, how long would it take such a treatment to actually take effect and for patients to be able to see an improvement or a change?
Yeah, that's a really good question. You know, the current thinking is that DUX4 is, you know, we know that it's expressed at low levels, even in FSHD patients. And we sort of assume that those, you know, based on some data, that it's expressed in sort of a dynamic fashion where, you know, the cells are all kind of poised to express DUX4 because they've lost this repression. And in some cases, they express a little bit, which causes some problems, but eventually the problems become so severe that the muscle cell dies. And then you get pathology. So yeah, if we were able to knock down DUX4 expression, presumably if the cell hasn't died yet and it's responsive to, you know, DUX4 repression, it should go back to being a healthy cell. So yeah, how long it would take, I don't know, but it'll be interesting to see the time course, you know, of functional improvement. In mouse models, of course, but yeah, definitely in patients, you know, beyond that.
So we've talked a lot about DUX4, but you also mentioned work on what lies upstream that might be triggering DUX4 expression in patients. Is there work being done to develop therapies that target those upstream triggers?
Yeah, so we're definitely working on that as well. So the more recent paper beyond this CRISPR work was identifying regulators of DUX4 expression. So basically, I did a small scale screen to identify new activators of DUX4. So not just the proteins that we're losing that we want, but what are these activating proteins that are actually turning it on that we want to actually get rid of. And so, yeah, I did find several good candidates. So we are currently studying those candidates in muscle, what they do, how important they are in muscle, and what the consequences are to inhibiting them, because these are great targets for drugs. to basically turn off the things that turn on DUX4. Yeah, absolutely. And other labs are also working on this.
Okay. There's a question about DUX4's role, again, in embryogenesis. So it turns on in the second day in human embryos. When does it get turned off?
I think shortly thereafter. I'm not 100% sure, but I'm pretty sure it really is on only at the cleavage stage, and then as soon as the cells are divided, it's off, and it stays off except in the germline, in the certain cells of the testes. So yeah, it's fascinating that this very powerful pioneer factor, that can drive these impressive embryonic programs of gene expression, and is absolutely required, early, but never again, and then when it is expressed, re-expressed, in the wrong time and place, it has such devastating effects.
Mm-hmm.
So I can also point to a really great paper that came out of Steve Tapscott and Robert Bradley's lab, showing recently in Developmental Cell that DUX4 is not just misexpressed in FSHD, it's also misexpressed in certain cancer cells, and it allows those tumor cells to actually escape immune surveillance. So some of the genes that DUX4 turns on at the cleavage stage are immune modulators, and it's thought that they protect the embryo from the maternal immune system. But cancer cells have found a way to turn DUX4 on and use it to their advantage. So some of these tumors actually, express DUX4, and DUX4 turns on these immune genes, and it allows the cancer cells to basically hide from immune surveillance. So that's actually another pathological effect of DUX4 being on in the wrong time in the wrong place.
Wow. Well, I am afraid we have reached the end of our hour, and we had no shortage of questions. I thank the audience for all of their questions, and I wanted to end with reading a comment from one of the audience. He says, "This is not a question, but a comment. Thank you so much for the hard work you and others do. I have learned so much today. I'm a father of a daughter who has FSHD. She's 28 and working 3 jobs. She never complains. Where can we donate just for the research for scientists like you?"
Oh, wow. Well, come by the lab anytime. We would love to show you the lab and take you on a tour of the area. And yeah, I mean, we have patients come by all the time, and it's really, really fun to meet people. And yeah, it's just a remarkable group of people that we're trying to help. And thank you. That means a lot to me and the rest of us.
Thank you. Yes. All right. Well, thank you so much.